Prior to their extinction, Neanderthals coexisted with humans as late as 30,000 years ago in Europe and Western Asia. This research explores the genetic similarities and differences in order to make estimates about the divergent schemes of modern humans and Neanderthals. Sequencing and Analysis of Neanderthal Genomic DNA by Thomas Baron. This study uses DNA extracted from a 38,000-year-old Neanderthal specimen in order to create a metagenomic library, which they designated NE1. Analysis revealed that only 2% of the library was contaminated with modern human DNA. 62,250 base pairs were recovered from NE1 using Sanger sequencing and massively parallel pyrosequencing. In order to test the pyrosequencing data quality as compared to the highly accurate Sanger method, 130 loci, or 6.2 kilobases, were compared. The two methods agreed at 99.89% of ungapped positions. These results were significant in showing the accuracy of the massively parallel pyrosequencing method. In order to determine if NE1 was a representative sampling of the Neanderthal genome, the sequences were scored against the human genome using BLAST-N. The distribution of the best hits in BLAST-N was then determined. This figure shows the amount of Neanderthal genome in red, aligned to each human chromosome in blue. The hits on the Y chromosome indicate that the specimen was male. As you can see in the graph, the amount of Neanderthal DNA aligned to human DNA is highly correlated to the length of the human chromosome. In order to date the human Neanderthal divergence, finding human-specific mutations was critical. When 37,636 orthologous human, chimpanzee, and Neanderthal positions were aligned, 34 human-specific substitutions were found. 171 Neanderthal-specific substitutions were also found. However, this may be due to the tendency of ancient DNA to undergo deamination of cytosine to uracil, resulting in high amounts of mismatching during both Sanger and pyrosequencing. Figure 4 shows the excess of C to T and G to A mismatches due to the deamination of cytosine to uracil. The data obtained in this study has allowed the first genetic comparison between humans and Neanderthals using nuclear genomic sequences. From the nuclear genomic sequences, it was calculated that the most recent common ancestor existed around 706,000 years ago. Figure 5a shows the relative log likelihood versus years to the most recent common ancestor. The 95% confidence interval allows a range of 468,000 years to 1,015,000 years. Figure 6 displays the diversion estimates for human and Neanderthal genomic sequences. To estimate the actual split time of the ancestral population that gave rise to Neanderthals and modern humans, a new model was developed, using the human and Neanderthal reference sequences along with 210 genotypes collected by the International HapMap Consortium. This explicit population model examined the relationship between Neanderthals and each HapMap population. The model assumed that Neanderthals and humans evolved from a population of 10,000 individuals instantaneously. A simulation-based composite likelihood framework was then constructed to estimate the time of the split. Using these models, the maximum likelihood split time estimates are 440,000 years for Europe, 390,000 years for East Asia, and 290,000 years for Yoruba. This averages 370,000 years. Previous research in a Neanderthal genetic contribution to the modern human gene pool is only relative to Neanderthal females and modern human males because of mitochondrial inheritance patterns. However, using the population models, the maximum likelihood estimate for Neanderthal contribution to modern genetic diversity is still 0. To make the research more specific, the potential to recover specific sequences from the metagenomic library was explored. To test the potential, 96 highly conserved mammalian sequences were targeted. PCR probes corresponding to the sequences were created and hybridized to PCR-amplified cave bear inserts. 5 target genes were recovered. When the method was tested on NE1, the Neanderthal metagenomic library, 29 of 35 sequences were recovered. This study revealed that Neanderthal genomic sequences can be recovered using metagenomic library-based approaches, helping to rapidly find genes of interest for recovery and analysis. The study also revealed that 99.5% of the recovered Neanderthal DNA is identical to modern humans. The research advances our knowledge of the evolutionary relationship between humans and Neanderthals. Future work will help us to better understand the profound phenotypical divergence between Neanderthals, the great apes, and humans.